this guy called adenyl cyclase, which is a protein / enzyme that's stuck within the membrane. And he does that. He takes ATP, which is an adenine guy, an adenine derivative, and he makes him cyclic into cyclic AMP. He turns on another protein called a kinase enzyme, a protein kinase, and then he creates changes in cell signals. So we're going to see this cascade in a lot of different areas, but this is what's known as secondary messengers, because the binding to the protein cell surface is what creates the cascade of effects inside. So thyroxine is the only one that is going to be different. We're going to look at him in a little bit, but most of all the peptide hormones are going to activate this way. So we're going to look at cyclic AMP. We're going to look at the G protein coupled receptors that activate cyclic AMP. We're going to look at G protein coupled receptors that activate this guy called phospholipase C. So he's just a different mechanism on the inside. And then we're going to look at guys called receptor tyrosine kinase receptors. And this guy, cyclic GMP, can happen just in and of himself without a G protein. So there's a tiny little example for that too. Okay, so G protein coupled receptors look like this. I want to show you this one first. So they are what's known as a transmembrane protein receptor, and they go in and out of, like a snake, in and out of the cell membrane like 7 times. So when you get your peptide hormone from the blood or the extracellular fluid and it connects, he creates changes within the enzyme, and then he's going to remember either turn on or turn off adenyl cyclase. So if we pop back to this guy, there's a couple of different pathways. So we can either activate him or we can inhibit him. So, and we can either activate cyclic AMP or we can activate somebody who's going to make our cyclic GMP, which we'll look at an example in a second. And then that's going to initiate reactions within the cell. And then the reactions within the cell are going to change the genes or change transcription, protein synthesis. And if we change the proteins that are in our cell, we change the function and what it does. So like one example is here. This is for catecholamines, adrenaline, noradrenaline, right, or epinephrine. So it might activate this guy here. This is a particular G-coupled protein receptor. And he, within your vascular system, for instance, in order for us, when we're in our fight or flight reaction, we have smooth muscle contractions, we have vasoconstriction, so we can get blood to the periphery in our brain and we don't pass out. So he's going the way of phospholipase C. So he's going to activate this guy. It's a lipase enzyme. He runs over and he finds this guy called phosphatidyl. It's inositol based. It's a phosphate inositol fat. He's going to cleave him up and create this guy called IP3. Then IP3 pulls calcium into the endothelial cells, and then we get smooth muscle contraction. So this is a G protein coupled receptor that activates phospholipase C. On the same speed, we can have a G protein coupled receptor that activates on the same neurotransmitter within the sympathetic nervous system, and now maybe this is for heart muscle contraction or the breakdown of glycogen, so we have sugar. And instead of activating phospholipase C, he's going to create cyclic AMP. He's going to have adenyl cyclase activate, make cyclic AMP, and then we'll have muscle contraction, glycogen breakdown, things like that within the cell. So this is what it would look like if it was inhibitory or stimulatory. So epinephrine activates our G protein. That turns on, so this guy was on here. He kind of breaks off this little alpha protein. This activation breaks this alpha protein off. He comes over here and he turns on adenyl cyclase. Now he's activated. He does ATP to our cyclic form. He turns on the protein kinase, and then the protein kinase then changes the cell's response. So this cascade would be stimulatory. This would be turning the cascade on. But say we want to inhibit it. So like If we wanted to stimulate it, maybe we pulled this guy off, which turned on adenylyl cyclase. If we wanted to inhibit it, maybe this guy gets pulled off. And you've left the adenylyl cyclase from working, and then you have cyclic AMP. So different hormones will act different with the different G protein coupled receptors. So in endocrinology and in PATH and stuff like that, probably more in AP3, you'll get a little more specifics into specific G protein coupled receptors, but this is the base of how they kind of work. This is the example of our G protein turning on phospholipase C. So he turns PIP into IP3. IP3 is what changes translation in the endoplasmic reticulum, and then we get a protein that changes stuff. We can also have just cyclic GMP just do stuff for us too, without a G protein coupled receptor. And the classic example is nitric oxide. So nitric oxide, as a very small molecule of gas, can just diffuse through the cell. So it can go, remember we saw that big picture in the beginning where nitric oxide was produced within the endothelial cell, a squamous cell, and then it could just diffuse over into the muscle, into the vasculature, the smooth muscle. Well, this is a picture of this. This is our endothelial cell. Nitric oxide was just made in the squamous cell. He comes over, and in the smooth muscle cell, he's going to then turn GTP into cyclic GMP. Then cyclic GMP turns that kinase enzyme on, which then creates the cell response. So that's not a G protein coupled receptor, but it's still a cell signal that'll create a change in us. This eventually, if we're using the idea between nitric oxide and vascular, this is going to create vasodilation. So this smooth muscle will relax in this response. Whereas the opposite was when we used adrenaline in the G protein coupled receptor, we had smooth muscle constriction. So the only guy who's really different is this thing known as a receptor tyrosine kinase. And he looks just like this, it's kind of funky, is that it's known as a dimerization of tyrosine kinase receptors. So that means two. So this is a tyrosine kinase receptor, and this is a tyrosine kinase receptor. And it's named for that because it has a bunch of tyrosines in it, and it has phosphates. So we're gonna end up popping some of those phosphates off, which is like a kinase reaction. So what happens we have our protein hormone come around, so that's the ligand, this guy up here, and when the ligand comes close to one of these guys are normally apart. And when the ligand comes in and he maybe goes to the one side, this guy comes close and he clamps next to him to hold the ligand in, and then we have this cross phosphorylation that holds the two together. And that creates a change in the protein all down through the cell membrane and into here. And then the phosphates are used as energy to activate this guy known as RAS. And he starts a bunch of kinase enzymes inside. So it's not cyclic AMP, not cyclic GMP, not IP3, it's a RAS activation, and he looks different. So, for instance, the tyrosine kinase receptors, insulin uses this. This is what insulin uses. When insulin binds, the tyrosine guy kind of clomps onto him, holds onto him, creates changes in the receptor, which activates RAS, which turns on kinase enzyme, which makes GLUT4 transporter vesicles come to the surface, so that now GLUT4 can bring sugar into the muscle and adipose tissue. Okay, so any questions on those? These are just kind of generic 4 different ways in which protein receptors can create a cascade inside. So to kind of reiterate, if you understand that there's 4 different kinds and can name them, that's helpful. So we have 2 G protein coupled receptors. One of them activates cyclic AMP. One of them activates PIP. We've got our cyclic GMP that can just happen usually by diffusion of some kind of molecule. And then we've got the last one, which is the tyrosine kinase receptor, which activates RAS. So if you can get just those couple 4 sentences down, I'm happy with you just knowing those pieces. Okay, so let's take a brief moment before we go on to talking about the thyroid. Revisit the idea about feedback mechanisms. So we talked about the hypothalamus, pituitary, and some kind of endocrine hormone feedback. Why do you go, okay, first of all, what type of control is that? Negative or positive? Like the HPA axis, the one we've talked about before. That's negative. Yeah. What are most homeostatic feedback mechanisms in our body? Negative, right? Yeah. What does negative mean? Describe that to me.
Whatever the product is, we'll go back and like slow down that whole process.
Perfect. Yep. If we have a lot of the downgrade product, we'll go back and slow down the top of the reaction. So sometimes we have small ones, like in all those allosteric enzymes we talked about in Biochem, we have little loop feedbacks. And then sometimes we have big feedbacks, like the hormone going back and talking to our brain, hypothalamus, or pituitary. So there's short and there's long loops. Okay, so it really controls the intensity of the signal, which is super important. But when we start to talk about the thyroid, when it comes to thyroxine, it gives us a nice snapshot as to whether or not the body overall is maintaining homeostasis. So if those anterior pituitary hormones are probably most important, but sometimes the hypothalamus hormones, with those being off, they give you a tremendous deal of look as to whether or not our body is regulating well. Okay. I'm just trying to, it's 9:30. It's 9:30. Do we need a break? Do you want a brain break? Do you want to get up and move around before we do that guy over there? That'll probably take about 20 minutes. Okay, let's do a brain break. So I saw that. Let's come back in 10 minutes. Is that okay? All right.